Okay. No, fingers crossed this works. Been a long time since I've done anything Zoom related. Tasha, could you do me a favor and stand up there so I can make sure I'm not missing your head? Cool. Perfect. Right, that's running. I think it's pretty much time to get going from what I can remember. Yep, that's good enough for me. All right. All right. Hi, everybody. Thank you very much for coming to, well, I mean, the grad students, they have to come to the talk, so, but thank you for coming anyway. And everybody else, it's my distinct pleasure to introduce Dr. Natasha Kirienko from Rice University. She is one of a very select group, tribe, cohort of people that use C. elegans to answer questions about immunity, pathogenicity, and basically use it as a model for asking really big questions about human health, right? Because no one cares about worms. They can kill millions of them, and no one bats an eye. And C. elegans is obviously a really powerful model, given that there's thousands of groups working on it, has its genome sequence, it has a whole wealth of tools available to it. And Natasha's going to be talking about some of those and the things that she's found along the way. So, hello, Mark. Come and begin.
Hello, and let's keep it all informal. At any moment, if something is unclear, please stop me and ask your question. When people ask me, what do you work on? I used to have a really awkward moment, because how do I tell in one coherent sentence that we work on cancer, neurodegeneration, post-pathogen interaction, and bacterial virulence factors? I not sound crazy. And then I found a way. Now I tell people, we work on mitochondria, because it doesn't matter what we do, at the end, it all comes to mitochondria. And one thing that gives me bittersweet feelings, and when you say mitochondria, you immediately hear, oh, the powerhouse of the cell. Well, that's true, but there is so much more to mitochondria. They involved in the maintaining levels of calcium and reactive oxygen species. They're important for lipid catabolism and lipid synthesis as well, participate in cholesterol biosynthesis, make decisions in life and death, not just apoptosis, but multiple cell death pathways can be activated due to mitochondrial damage. And via modulating level of reactive oxygen species, mitochondria helps to make a decision whether the cell should differentiate or maintain its stemness. Without mitochondria making energy, cell can get by. They called raw negative cells. They can exist. We can make them in cell culture for mammalian cells. We can make it for yeast. They're not happy, but they can survive. If you remove mitochondria as an organelle, eukaryotic cell will die. So yes, energy production is important, but there is more to mitochondria, and I'm going to talk a little bit about it during my presentation. Originally, when I was thinking of what should be my independent niche for research, I was interested in studying post-pathogen interaction. Infectious diseases still a huge burden to our world, both emotional and societal economical. About 50% of all premature death is still due to the infection diseases, which happens at the time when we're making such a huge progress at performing organ transplantations or fighting cancer. And there's still nothing we can do to those little bugs that were infecting human beings for thousands of years. Unfortunately, in part, this is due to the rise of antimicrobial resistance, or the ability of bacteria survive on antimicrobial treatment. And Currently, well, that's from back to the data from about 2018-19. There are 700 cases of bacterial infections a year were detected. Now the number is much higher. Recent statistics reports that there are about 2.8 million of such infections a year, and even more worrisome, by 2050, the number of infections with antimicrobial resistant bacteria is expected to reach about 10 million, surpassing that of cancer. That's the number of deaths. So for this reason, antimicrobial resistance is a world level crisis. As I mentioned, the reason for it is that bacteria can develop resistance to antimicrobials. And the level at which it happens is completely astonishing. So on the top, you can see colors, different colors to represent when different class of antimicrobials was introduced into clinic. And in the bottom is when resistance was detected. So you see that the line in the bottom is somewhat shifted compared to line in the top, but not that much. It only takes a year or two for resistance to be spreading in the population of bacteria. Sometimes it's few months. Occasionally, resistance can be detected even before widespread use of antimicrobials. And that's the problem. What's the point for a company to develop a drug, spend 20 years and couple $1 billion to put it on the market, and couple years later it becomes obsolete because resistance develops? Another way, if we want to keep resistance from developing, new drugs can be used as last resort molecules, only used if nothing else helps. But then we're not selling it widely, so company is not making money regardless. And for this reason, the number of antimicrobials that being developed been steadily decreasing, while antimicrobial resistance was on the rise. And these just economical problems, they also chemical problems, as we use lots of obvious chemical scaffolds as a base for drug design. So it's harder and harder to come up with new chemistry. And biologically, we already thought of great targets to use to kill bacteria: replication, translation, integrity of bacterial membrane. So and bacteria find out a way to defend against it. Coming up with new targets is also hard. So then what else can be done to solve this huge problem of antimicrobial resistant bacteria? One approach that my lab is interested in is not to try to kill bacteria, but instead inhibit its virulence. If you think of a pathogen, it has lots of different types of arbors that it's using to damage or kill the host. It could be formation of biofilm, usage of secreted toxins, presence of worm sensing signaling that coordinate bacterial behavior. So to help the patient, we don't need to kill the pathogen. When we're trying to kill bacteria, it fights back. But what if what we do instead would be simply disarm it, to force bacteria to go into a commensal-like state, to be just like one of those millions, hundreds of millions bacterial cells that already live inside of our body as part of microbiome? Then bacteria is still there, but it does no harm. And eventually, this pathogen would be outcompeted by commensals that much better adapted to living inside of our bodies. So sounds like a promising approach. Unfortunately, to use it, we need to understand what are important virulence factors that we need to block bacteria from being able to use. So for that, we need to understand host-pathogen interactions. And to study host-pathogen interactions, my lab uses mostly model nematode Caenorhabditis elegans. Worms are small, they're only about 1 millimeter long. So here you see a picture of worms inside of 384 well plate. The whole plate is only this big, and we can stuff 8,000 worms inside of it. They're quite comfortable. So it really facilitates any kind of high throughput studies. Importantly for us, C. elegans have powerful genetics with lots of mutants or RNAi constructs available if you want to study certain pathway or certain genes. It's evolutionarily conserved, including innate immunity or host defense pathways. And it's transparent, so we can easily monitor pathway activation. But the important feature, most important, I think for last, it's bacteriavore, meaning it's bacteria. Normally, we see that E. coli, non-pathogenic bacteria, Escherichia coli, and their worms live only 2-3 weeks. That's their normal lifespan. They only take 3 days to go egg to egg, very fast. If instead of E. coli will be mean scientists and will give them a pathogen, then these pathogens would infect worms and they will die much faster, from several days to a bit over a week. A number of human pathogens, including gram-negative bacteria, gram-positive bacteria, or fungal pathogens, can infect and kill C. elegans. Importantly, virulence mechanisms they use are the same that when they infect human beings. So when we use C. elegans, we can study phenomena that are relevant during patient infection. A lot of assays that were done using C. elegans were done on agar, where you put pathogen, put worms on top of it, and every day you look at them and ask, are you dead yet? And record the numbers. You can do it, and it can give you lots of data and lots of hours undergraduates spend staring at the microscopes, or unlucky graduate students. But you can also use alternative approach and try to automate things. In order to reach higher throughput, we switched from agar to liquid-based assay, where we similarly let worms grow on their favorite food, E. coli, and then we use special machine that costs ballpark like a house that can put worms into creative oval plates, exact number of worms of exactly the size we want, or with the fluorescence we want. So this way we have a very uniform population of worms. We can add pathogen, or we can add different bacterial mutants, or we can add pathogen and drugs, depending on what we want to do. And then we incubate this plate with host and pathogen for appropriate lengths of time to ensure host death. Then we remove pathogens by washing the plates and use a special dye that stains only dead worms. Plates are imaged on automated microscope, and that's what our images look like. If worms are alive, virtually no staining is observed. If worms are dead, even in bright field, you can see the difference in their shape. Heat rods versus moving curves. However, the difference in fluorescence counterparts is even more dramatic. Nearly 100% of fluorescence versus ballpark 10% background. We can analyze these images quantitatively, and for each of them, based on the fractions of fluorescence, infer fraction of death. And this way, it's unbiased and eliminates human error. So, if worms are fed E. coli, that's your happy worms that stay alive. Pseudomonas aeruginosa, dead worm. And we work with this type of images a lot. After the assay was developed, I was curious to see how does bacteria kill the host, because Pseudomonas aeruginosa is particularly known for having multiple virulence mechanisms that can be activated during different phases of infection. So we wanted to see what's happening here. In order to do that, we use different mutants in Pseudomonas aeruginosa and check which of them have compromised ability to kill the worm. So each individual image here represents a well with about 20 worms, but we can only see those that dead. Worms fed E. coli, pretty much no dead worms. Worms fed with wild type Pseudomonas aeruginosa, majority of them dead. We used mutants in biofilm, in production of toxic molecules phenazines, mutants in cyanide production, and worms remain dying at the same level as wild type Pseudomonas aeruginosa. However, when we use mutants in pyridine biosynthesis, we notice that the number of dead worms significantly decreased. So what is pyridine? And that was the first time when I learned about it. So here's its structure, just to show you that it's made non-ribosomally. It has some non-standard amino acids present in it, like, for example, hydroxyornithine or D-serine, and it has chromophore, so it's fluorescent. This molecule's job is to go into the environment, find ferric iron, iron 3+, and bring it back to bacteria. Ferric iron is critical micronutrient, and it's the iron itself. It's most abundant element in the Earth's core, or one of the most abundant, but it's always complexed with something. So it's not biologically available. That's why living organisms really need to find a way to obtain iron. Or during the infection, we as mammals have our own iron stored, securely bound to proteins. So pathogens also have hard time obtaining iron during infection. And that's why they use molecules called siderophores, which means iron carriers, to bring iron to them. So pyridine is one of the siderophores. And if we use mutants in pyridine synthesis, we see that C. elegans death is now much lower. If Pseudomonas cannot make pyridine, it's much less efficient in killing C. elegans. And that's just to show that pyridine is fluorescent, so we always know if it's made or not. And usefully for us, it's only fluorescent when it doesn't bind iron. Iron is carbonated between these 6 oxygens, so when it's bound, chromophore structure changes and fluorescence is quenched. There was quite a bit of literature on pyridine, because without siderophores, bacteria cannot obtain iron, which means it cannot grow. That's why siderophores and pyridine were known to be important for virulence indirectly, mostly as providing nutrients to the bacteria. Also, pyridine, when it binds to iron, was known to activate a number of nasty toxins, including proteases that Pseudomonas makes that just breaks phosproteins, or exotoxin A, which is a potent inhibitor of translation. So previous knowledge on pyridine, important for Pseudomonas virulence, likely indirectly through controlling nutrients and other virulence factors. We wanted to see how important pyridine is overall, because Pseudomonas makes number of siderophores. Pyridine is major one, but pyochilin is another one. So we wanted to see how important is the presence of pyridine in the context of infection. Through our collaborator, we obtained a panel of 69 Pseudomonas aeruginosa isolates from a sputum of pediatric patients with cystic fibrosis. And we noticed that of these 69 isolates tested, black line is our lab strain. About third were making even more pyridine than lab strain, and overall 2/3 were making appreciable amount of pyridine. When we analyzed the ability of these 69 isolates to kill C. elegans, what we observed was that there is a correlation, negative correlation between C. elegans survival and the amount of pyridine. The more pyridine is made, the fewer worms will survive. So looks like indeed, so far, what we find is consistent with the literature. Pyridine is important for host killing. And other research also confirmed that pyridine can be identified in a majority of patients' sputum. So it is a very common factor in chronic infection of Pseudomonas. However, I wanted to explore whether pyridine may have more direct role in virulence. In order to do that, we produced bacteria-free cultures that will contain pyridine. If we incubate worms with the media, these are black circles, all the worms alive. If we make these cultures, these supernatants, from E. coli culture, where pyridine is not made, then worms alive again. If we prepare supernatant from Pseudomonas culture and have high levels of pyridine, worms die even in the absence of live bacteria. If we add iron that will naturally bind to pyridine, likely blocking its toxic effects, worms remain alive. So pyridine may have some direct toxicity to C. elegans that's stemming from its ability to chelate iron. And we confirmed that by obtaining commercially available purified pyridine, there are similar things. When we mix pyridine with C. elegans, we observe worm death. If we add iron exogenously to bind to pyridine, then worms remain alive. Pyridine is cytotoxic, but why? Well, first, we hypothesized that toxicity of pyridine has to be related to its ability to bind iron. Iron is important not only for bacteria, but also for us and for C. elegans. There are two sources of iron, either media or the host. And we hypothesized that depleting iron from the media will be likely not good for C. elegans, but it will take days and, well, weeks for worms to be affected. And here we see worms dying within 24 or 30 hours. So we thought in order to do that, pyridine should be affecting host iron. And since it's a chelator, to affect host iron, it needs to be inside of the host. To test this hypothesis, we took advantage of pyridine's innate fluorescence and we stained C. elegans gastrointestinal lumen with the red dye. So this is C. elegans mouth and pharynx, and if filtrate or supernatant was made from wild-type Pseudomonas containing pyridine, we see lots of pyridine fluorescence within the worm. If we prepare analogous material from pyridine biosynthesis mutant, that no pyridine signal is observed, or if we add iron natural pyridine quencher, same thing, no pyridine fluorescence. And we can quantify host iron using either type of spectrometry or some other biochemical assays. And indeed, we observe that in the presence of pyridine, host ferric iron significantly drops up to about 40%. So likely this loss of critical micronutrients leads to aberrant biochemical reactions inside of the cell and then host death. You have a question?
Sure. So the pyridine is clearly getting into the animals. Is it a active uptake? Is it damaging the cells and then getting absorbed?
I will have a little bit on it later. And by the way, I don't have any way to track time, so let me know when it will be 12:50. So we hypothesize that pyridine gets inside and causes some kind of havoc. But what cellular processes organelles will be most sensitive to pyridine? And that's where we thought of mitochondria, because inner mitochondrial membrane is 80% proteins. Majority of proteins there are electron transport chain complexes, which have abundant iron sulfur clusters. And iron sulfur clusters will have iron that pyridine would like to steal. So we looked into mitochondrial function first. Electron transport chain complex 1 oxidizes NADH to NAD+, and then pair of electrons is donated to electron transport chain. We had a reporter that will be fluorescent when bound specifically to NADH. Normally in the cell, NADH should be quickly oxidized and pick electrons again in the citric acid cycle and carry them to complex 1 and so on, but basal level of NADH will be low, virtually no green fluorescence. However, if we add pyridine, we see strong decrease in GFP signaling, suggesting that a function of complex 1 is inhibited and we have buildup of NADH. Mitochondrial quality control is important for the cell, as less energy means cell will not be healthy. And usually, if mitochondria have low level of damage, it would be diluted with healthy mitochondria. It will fuse with healthy mitochondria to dilute the damage. However, if there is high level of damage, if it's mitochondria losing its membrane potential, then it's targeted for recycling using process called mitophagy or autophagic degradation of mitochondria. So then this mitochondria will be engulfed and taken apart. So we looked at what happens to mitochondrial network in the presence of pyridine. When worms fed on E. coli that doesn't make pyridine, then in their muscle cells, mitochondria make this beautiful network of long branch tubulars when they connected to each other. If we add pyridine, sometime later, we notice that networks just fall apart and we're starting to see fragmentation of mitochondrial network, and formation of puncti that likely are autophagic granules formed. The similar phenomena is observed if instead of pyridine, we will use phenanthralin. It's a chemical iron chelator with totally different structure, but common biological function. So indeed, iron chelation damages mitochondria and triggers autophagic recycling of mitochondria. We did a number of assays to show mitochondrial function decrease. For example, here we took advantage of PINK1 GFP translational fusion. PINK1 is a kinase that activates mitochondrial autophagy. So usually if you have high level of PINK1, that means mitophagy will start. That's why normally you have very low level of green fluorescence, and red is constant, that's our normalization control. But in the presence of pyridine, you see high level of green. So iron chelation initiates programmed recycling of mitochondria, which leads to the decrease of in mitochondrial to nuclear genomic ratio. So we have less mitochondria confirmed by the lower mitochondrial DNA. And of course, if you have less mitochondria, you will also have less ATP. So presence of pyridine is capable of causing substantial mitochondrial damage that has strong negative effects on overall cellular health. But that's all C. elegans. How is it relevant for mammals? To do that, to investigate whether it's relevant, we started with macrophages. So these are macrophage cell lines, and when we treat cells with pyridine containing filtrate, we see strong decrease in macrophage survival. If analogous material was made from pyridine mutants, then macrophages survive much better. So blue stains any cells and red stains dead cells. Much more cell death in the presence of pyridine. Similarly to C. elegans, we wanted to see, does pyridine get inside of mammalian cell? Because with cell culture, we likely have individual cell resolution. If we incubate macrophages with pyridine, we can observe pyridine fluorescence inside, absent in pyridine deficient mutants, absent if we add iron. However, gallium can also bind to pyridine, and unlike iron, gallium makes it even more fluorescent. So if we add gallium, we have even brighter fluorescent signal. And all of these data together confirm that signal we see indeed is from pyridine, and it can get into mammalian cells. And one of the reviewers asked us, what if it's only sitting on the membrane and removes iron that leaves the cell, or iron from the lumen? So thankfully to confocal microscopy, we can answer that question as well. We stain macrophages and their nuclei in green, pyridine is in blue, and the cellular membrane is in red. So here, especially at larger magnification, we can clearly see pyridine being sequestered in the cytosol of the cell, and we don't have anything of a kind in a pyridine deficient mutant. So indeed, pyridine gets into mammalian cells. And of course, next logical step to see what it does to mitochondrial health. When it comes to the shape of mitochondrial network, I hope you agree that C. elegans are superior to people because you don't see this neatly organized network here. It's more of a chaos of these long tubular forms that they make. Usually these are made from several individual mitochondria. If we add pyridine, however, we see breaking down of this structure, so oligomers of several mitochondria. Now we predominantly have punctine. And if we're adding material from pyridine biosynthesis mutants, mitochondrial structure is not significantly affected. Everything we saw in work is confirmed in cells, including strong correlation between survival of macrophages and pyridine production. The more pyridine is made, the fewer macrophages will survive. And I was interested in macrophages because Pseudomonas is quite common in pneumonia or lung infections in patients with cystic fibrosis, and macrophages are immune cells that will be fighting with bacteria there. But of course, other relevant cell type would be epithelial cells. So we looked at the effect of pyridine on epithelial cells. Normally, this will make nice monolayer, and here we stain the cell membrane so you can see all the junctions between cells. If we add pyridine, monolayer breaks down, and we're starting to see these big gaps in the structure of epithelial monolayer. And the damage is much less if we use material from pyridine deficient mutants. So unfortunately, this commonly produced siderophore is damaging to both types of cells it will encounter during lung infection. And as a final step, in a collaboration with Dr. Carolyn Cannon, who is from Texas A&M, but College Station 1, we did experiments in mice. Based on our C. elegans research, we chose 4 strains of Pseudomonas, 2 high pyridine, high killing of C. elegans, and 2 low pyridine, virtually no killing of C. elegans. And these 4 strains were tested in mice. Strains that kill worms also killed mice, and we were able to detect pyridine being produced within mouse lung, confirming our model. So this is how, starting from C. elegans, you can identify a virulence factor that's important in mammalian infection. And something I'm not going to talk at all about today is we did a number of drug screens to find molecules that will inhibit pyridine. And some of those are also capable of rescuing animals, including mice during infection. But that's what bacteria does, trying, and it's not even, I don't know if it's trying to kill the host. All it does is trying to get iron and then causing this kind of damage. But what do hosts do during that? Are they just sitting and waiting to die, or are they fighting? To answer that question, we analyzed transcriptomes of C. elegans that were infected with Pseudomonas producing pyridine, condition 1, or any other type of infection, which would be numbers 2 through 11, like infection with Enterococcus fecalis, Staph aureus, Candida albicans, feeding on E. coli, and all the other conditions. We were interested specifically in genes that will be upregulated only when pyridine is present, and that was cluster 1. Cluster 1 contained about 55 genes and was very specifically up on pyridine only. We hypothesized that if these genes are regulated in the presence of pyridine, there has to be some transcription factor that upregulates them. So we wanted to find the putative binding site to that transcription factor. We looked at the promoters of those genes and noticed that this motif, TCT, GCG, TCT, TCT, is strongly enriched. It was present in about 40% of genes in the cluster, and it was enriched overall in Pseudomonas that makes pyridine. Well, enriched in C. elegans infected with Pseudomonas that makes pyridine. If Pseudomonas doesn't make pyridine like under other infection, then we do not see motif enrichment. Seem to be pyridine specific. Next, we wanted to know what does activate it, because turns out this motif was published before 7 times by 7 different labs. 6 happened to be C. elegans and 1 happened to be mice. Originally it was found to be upregulated by alcohol acute ethanol stress, and that was the study that also found it in mice. But it was later shown to be upregulated by strong heat shock. We showed that it can be upregulated by iron chelation, not just by pyridine, but also by phenanthroline, oxidative stress caused by juglone, and some others. So what's common between all of these stressors? My hypothesis was likely mitochondria, because it's at the center of everything, right? To verify it, we took bona fide mitochondria damaging drugs like complex 1 inhibitor rotenone, complex 3 inhibitor endomycin A, and so on. And each of these mitochondrial poisons was able to activate the element GFP reporter, which we called 3xS GFP, because we have the element 3 times in front of GFP. So looks like mitochondrial damage activates S3. But mitochondrial damage can come from different sources. Problems with mitochondrial transport, deletions in mitochondrial DNA, accumulation of reactive oxygen species. So as a lot of the stressors, including malfunction of electron transport chain, may lead to higher level of reactive oxygen species, we wanted to test this hypothesis. We exposed worms to either rotenone or rotenone plus N-acetylcysteine, and we noticed increase in ROS. If we add something like a reactive oxygen species quencher ascorbate, we see S3 fluorescence decreasing and staining for reactive oxygen species, dihydroxyethidium, DHE, also decreasing. So we had a very good correlation between level of ROS and fluorescence of S3. To finally confirm it, we used the ROS scavenger sample that specifically removed ROS in mitochondria, and we noticed that if we combine rotenone, S3 goes up, ROS goes up, but in the presence of sample, we see decrease in fluorescence of S3 and decrease in ROS. So what happens upon pyridine exposure is increase in reactive oxygen species, and that increase is sensed, leading to the activation of a S3 pathway that likely recognizes superoxide as that's the type of reactive oxygen species that dihydroxyethidium states. So taking together, Pseudomonas makes pyridine that gets inside of C. elegans, steals host iron, including that from mitochondria. Mitochondrial damage leads to activation of reactive oxygen species. It also leads to reductive stress. Then unknown signal transduction cascade is being triggered, which leads to the expressions of genes that have S3 elements in these promoters. Some of the S3 genes are chaperones, helping to fold proteins, and some other genes encode for proteins that involved in mitochondrial repair. So what I told you so far is mitochondrial damage is bad. It can kill cells. How can we take advantage of it? Well, turns out sometimes we need to kill cells, and that's when the cells are cancers. But cancers are different. So you can't just think, hey, let's kill mitochondria of cancer cells as it will always work. First, we wanted to know, are there any even relationship between cancer and mitochondria? To answer this question, we started with bioinformatics. National Cancer Institute has a panel of 60 cell lines called NCI60 that represent 9 different types of cancer. So we obtained the data available for the cell lines, including sensitivity to different drugs. We choose 14 drugs that damage mitochondria and 14 drugs that do not damage mitochondria, like microtubule inhibitors, for example. And then we looked at the correlation between parameters like age or sex of a patient or type of cancer and sensitivity to mitochondria damaging drugs versus control drugs. Only one significant correlation was discovered between type of cancer and mitochondrial damage. When we ranked these 60 cancer cell lines, with 1 being most sensitive and 60 being most resistant, what we noticed, hit number 1, leukemia, hit number 2, cell line, leukemia, number 4, leukemia, number 7, leukemia as well, starting to see the pattern, right? And if you look at the very bottom of the list, it was most resistant to mitochondrial damage. There you'll see ovarian cancer. So it looks like, at least for some cancer types, they will have a very specific pattern of either sensitivity or resistance to mitochondrial damage. With leukemia, if we're averaging rank of the 6 leukemia cell lines present in the panel, we'll get 5.5. Very low, non-random distribution, suggesting that if we want to target mitochondria in cancer, that we have our guide. Those are leukemia as most sensitive to mitochondrial damage. To confirm this experimentally, we obtained a panel of acute myeloid leukemia cell lines, highlighted in red, and compare their survival on mitochondria damaging molecules like mitoxantrone, doxorubicin, actually standard treatment for leukemia, and uncoupler CCCP. We noticed that in every instance, leukemias were more sensitive to these molecules than healthy blood cells, peripheral blood mononuclear cells, or PBMC. This will be my control for the rest of the presentation. Glioblastoma U251, or ovarian cancers that were predicted to be resistant. They resist. And here, just to show you how some of the assays work, live cells are green, dead cells are red. You treat with mitochondria damaging drug and see large fraction of dead cells. Damaging mitochondria is bad, but how can we make sure that we affecting healthy cells less? Cancer cells have crazy upregulated metabolism. They're known to significantly upregulate glycolysis, which is called Warburg effect, and they also need more energy from mitochondria. So in leukemia cells, there are many more mitochondria than in healthy blood cells, but these are less functional. And I don't want to bother you with biochemical details. Just overall, this large number of mitochondria in cancer cells works pretty much comparably to the small number of healthy mitochondria in healthy blood cells. So we thought that what if we deliver a 1-2 punch? What if we block ATP formation in mitochondria using uncoupler CCCP, and we block glycolysis using 2-deoxy-D-glucose? Then cancer cells would have much more severe damage because its mitochondria are less functional, and they would be much likely to have to undergo cell death after this treatment. So indeed, when we combine 2 drugs, we observed in our synergy plot a lot of area in red, which means synergistic interaction. Green means antagonism and clear color neutrality. So this synergy was specific to leukemia cell line OCI AML2, but was not observed for healthy cells PBMCs. And the difference in synergy led to difference in survival. We have about 15% surviving leukemia cells and much higher survival of healthy cells. Overall, this hypothesis that we can kill leukemia cells using mitochondrial damage and even do better, but combining it with another drug works. But can we even push this difference in killing more? To address this more systematically, what we did was choose 6 mitochondria targeting drugs
some of which are known to be used in leukemia treatment, like EBT, cytarabine, ABT 199, and IAPS 10759, and 6 complementary drugs that do not target mitochondria. 6 times 6 led to 36 combinations. Of these 36, we observed strong synergy in 22 of them, which is a very high number. If you start randomly mixing drugs, you will not get that. And importantly, of 22 synergistic combinations, 21 were selective. It means they will kill leukemia AML cells at much higher rate than healthy cells. An example how it looks, the drug combination synergy plots in cancer cells, high synergy lots of red, and in healthy cells, no synergy observed. And that's different in survival. So right here, we have less than 10% of cancer cells surviving and over 95% of healthy cells surviving, suggesting that this type of synergistic combination may lead to decreased side effects, which is very important when you're treating elderly patients. So, mitochondria are the target for, at least for treating leukemia. What can we do next? Well, the molecules that currently used in leukemia treatment do target mitochondria, but what I call non-specifically. Cytarabine is a cytidine analog that can easily target nuclear DNA. It's just harder to get into nucleus than to get into mitochondria. And same for doxorubicin, it's a topoisomerase inhibitor. So yes, they do preferentially target mitochondria, but they were not designed for that. It just randomly happened, and it's not, the differential is not that big. What can we do to target something mitochondria specific? And that's where we thought of mitophagy, autophagic degradation of mitochondria again. And that's the kinase PINK1 I mentioned in the past that can trigger autophagic degradation of mitochondria. This time we did a drug screen where we looked for small molecules that, just like our control, sodium selenide, can activate mitophagy. And that's just how control screening plays used. If we use vehicle control, virtually no green fluorescence. If we activate mitophagy, then we see high level of GFP, and we have clear separation for this population, meaning we should be able to identify strong and moderate hits easily. We screened about 45,000 small molecules, and from these only found 8 that were able to stabilize PINK1, increasing its fluorescence. So if you see PS molecule, that's for PINK1 stabilizer. And that's an example of one of our hits, PS155, which causes green fluorescent signal from GFP. And if we use worms without fluorescent reporter, then there is no random green absorbed. So that green fluorescence is mitophagy or PINK1 specific. We identified those molecules in C. elegans, and we tested them in C. elegans. Indeed, we observed that all of these molecules activate fragmentation of mitochondrial network, which is a hallmark of mitophagy. And they also usually decrease either mitochondrial mass or mitochondrial membrane potential, and occasionally both. And here we can even see formation, some examples of autophagic functi. So indeed, we find the molecules that doing what we expected them to do, activate mitophagy. But our goal was to find new treatments for cancer. So let's take them to cancer cell line. Out of 8 molecules identified in C. elegans, 6 were able to a different extent to kill leukemia cells. For these 6 molecules, we obtained a number of analogs, I think overall about 100 molecules total. And we tested these analogs for the ability to kill leukemia cells MOM13 versus healthy PBMCs. LD50 is the concentration you need to kill 50% of cells. We noticed a number of molecules that were active in micromolar or even nanomolar range. And for a large number of them, you need at least order of magnitude more or 24 times more drug to kill healthy cells, giving us a very good differential. So here I showed our 3 most promising molecules that we wanted to explore further. And that's an example of cancer cells versus healthy cells, so you see difference in killing. This is a structure of our candidates. Well, they're not particularly meaningful, but you know, I just don't want to give you an idea that they're very secret and we're hiding them. What's important is these molecules, at least the PS127 family that came from original PS127 hit, they were able to activate mitochondrial growth to a high level, only in leukemia cells. Because healthy cells have healthier mitochondria, they harder to trigger growth. So here we see no difference in reactive oxygen species in healthy cells, but very high growth in cancer cells. And this activation of ROS is important for killing cancer cells. If we add ROS scavenger and acetylcysteine, then we decrease ROS and increase cancer cell viability. So that ROS production is one of the mechanisms for triggering cell death. Importantly, just to see the effect on cancer versus healthy cells, we measured ATP levels. And we saw a significant decrease in ATP formation in cancer, but not in healthy cells, with the exception of a single molecule that we immediately deprioritized. So indeed, this chemical structure of drug family specifically targeting cancer cells mitochondria. Moreover, they're also capable of synergy with existing leukemia treatments like IAX 10759 that targets complex 1 and doxorubicin. And this synergy is cancer specific. We do not observe large red peaks in healthy cells. The difference in synergy leads to a difference in viability. We see less than 30% of cancer cells surviving, while more than 80% of healthy cells. But these are just combinations that hasn't been fine-tuned. They just give us an idea that we can combine these drugs and effect would be beneficial for selectivity. We confirm all our findings in primary cancer cells that we get from patient samples. Immortalized cell lines can be a bit different. But indeed, for patients, you have much more variability because you don't know which other mutations present in their DNA, so it's less uniform than with cell lines. But still, in some cases, we have a dramatic difference between cancer and healthy cell survival, but we always have at least 50% difference in the outcomes of the treatment. Well, the next logical step after patient cells would be mice. This was done again with collaboration with another lab at MD Anderson Leukemia Department, Marina Konopleva's lab, where they engraft mice with leukemia cells that had luciferase in them, so we can monitor propagation of cancer by measuring luciferase luminescence. And these mice were treated with our compound. So cancer cells luminesce, so you see them here as a tumor form, and tumor formation was significantly attenuated upon drug treatment compared to untreated controls. And also when we treated mice with this drug, we noticed significant increase in their survival, even when drug amounts used were very low. Here we're talking about sub-micromolar or here 5 milligrams per kilogram. So indeed, these molecules could be promising leads for developing of future drugs that target leukemia. And today I told you about lots of different things that all related to mitochondria. The bacteria make special toxins, siderophores, that can target host cell mitochondria. And hosts respond to that by activating mitochondrial defense pathways, including S-REA. And this pathway is sensing ROS that is released by damaged mitochondria. Mitochondria can be used as a target to kill leukemia cells, specifically if you combine mitochondria targeting drug with another molecule with different mechanisms. So with that, I want to thank my lab who did all of this research, and that's my PhD advisor who's been visiting us on that day, so he's not in my lab, and our funding sources. Thank you for your attention. Yes, please.
The mission of that kind of product and that is more environment,
The answer will be different depending on model. In C. elegans soma, necrosis is pretty much the only type of death. Apoptosis happens in C. elegans, but only developmentally. So in that case, yes, guaranteed necrosis. In mammals, it's hard to answer. Lots of cell death assays like Alamar Blue, XTT, MTT, they measure mitochondrial function. So the cell death would be equated to mitochondrial damage. That's why we usually use nuclear staining, but then it's harder to understand. We try inhibiting apoptosis or ferroptosis and don't see much difference. So it's likely necrosis / necroptosis. Yeah, so far we use caspase inhibitors like Zivad or the caspase 1 inhibitor. Didn't see much effect. We did not do Westons for individual caspases. What we looked for inflammation activation, so we see at least some inflammasome activated caspase going up, and NLRP3 goes up, and some of the interleukins go up as well.
So I had a couple of questions in case anybody else had them. So essentially the mechanism of PVD toxicity is its binding of iron. But earlier on you talked about how iron-bound PVD also activates other processes. Yeah. So is the mechanism of PVD killing purely, do you think, in terms of your model at least, to its iron sequestration, or might there be some other kind of funkiness going on as well?
In C. elegans, likely mostly iron sequestration, because worms are not good at storing iron. They have about 10 times less iron than mammalian cells, so they'll be very sensitive to it. So they are very, very sensitive to killing by just filtrate that will contain pyruvate, but not other factors.
Mm-hmm.
In mammalian cells, iron storage could be higher, and there are also more mitochondria. Like macrophages will have hundreds of mitochondria in the cells, while in C. elegans it's more like 20, 30. So then pyruvate can get more iron, will get back, stimulate production of more virulence factors. So then these virulence factors will start to match up.
So is that why even getting rid of pyridine doesn't decrease death to zero?
Pseudomonas makes a lot of virulence factors. It's amazing for me to see that, well, in cell culture, filtrates material produced from pyruvate mutants kill to, let's say, some extent, but in mice, mutants that don't make pyruvate don't kill. So it depends in vivo or in vitro, because when we're talking about in vivo infection, there's also role of host immunity. So absence of pyruvate attenuates infection a little bit, and then immunity can clear it. When we try to culture bacteria from lungs of infected mice, we can get wild-type Pseudomonas, but we can never get pyruvate mutants because they clear the way. So even that little advantage in killing can go a long way when host-pathogen interactions are really well matched.
Also, one last question. So in the later work that you showed about the drug development,
and different compounds that you selected. In some of your controls, there's still there's obviously a large gap in viability between your controls and your combinations, but some of the controls still had, say, 80% viability. Is that just a normal consequence of doing that work, or is that suggesting there's still some activity like in your
usually if you have 80+% survival of healthy cells, that's considered acceptable, because everything is damaging. And another longer answer to that, we find the molecule from primary heat of 50,000 molecule library, which is not that great. Yes, we ordered about 15 or so analogs from that class, but again, that's not that many. So now that we know that the structural class is really promising, we did in silico analysis when we screened the 4.2 million small molecules for specific functions that predicted 2 PS127 class molecules, and then we found 95 more of them. So among those 95, there easily could be better molecules. And if I'll get funding to continue that work, hopefully, then we really may be finding a future promising leukemia treatment.
So it's like an iterative process. So from that 95,
Correct.
you get better predictive power.
Yeah, and given that those 95 came from 4.2 million, I don't think you can do much better than that. Yes?
Are there any other situations besides using this process for targeting cancer cells that we might consider using, like purposely damaging mitochondria for?
So damaging mitochondria a little bit can be good. There's a thing called hormesis. You know, what doesn't kill us makes us stronger. So if we'll use a little bit of mitochondrial damage, I'll compromise electron transport chain slightly. That leads to long living. And that's something what dietary restriction does. It just slows down all the processes, slows down electron transfer, and that's less loss and less damage, longer lifespan. So a little bit of mitochondrial damage, if it will slow down electron transfer, could be beneficial. Certain mitochondrial mutants live longer. Mitophagy itself is even more complex because it happens in different rates in different cells. In our neurons, for example, mitochondria are particularly important as neurons rely on glucose. They can't efficiently process fatty acids using beta oxidation, so mitochondria and oxidative phosphorylation is a big deal there. But neurons are also very bad at turning over mitochondria, and that's a problem in neurodegeneration. Like I talked about PINK1, another mitophagy protein, Parkin. It's called Parkin because it's found as complicated factor in Parkinson's disease. If you have mutation in mitophagy, then there are more severe and earlier onset of different types of neurodegenerations because of accumulation of damaged mitochondria. So our other hypothesis was if we'll stimulate mitophagy just a little bit, not to cause a lethal threshold, that may be beneficial in neurodegeneration because it will help them get rid of damaged mitochondria. And indeed, some of the molecules were not great mitophagy activators, but because they were so weak, they were helpful to provide rescue in three different neurodegenerative models in C. elegans: Alzheimer's, Parkinson's, and Huntington's disease. So that's another venue we're pursuing in the lab. Now we have, we're working on a contract with pharma company that will give us 250,000 small molecules to screen for mitophagy activation. And they're specifically interested in neurodegenerative applications. So if we'll find any new molecules that fight cancer, that's our own bonus.
I have the for
PINK1 is a kinase that activates mitophagy. So if we can stabilize it or just express more of PINK1, have more of PINK1 protein, that will likely to trigger mitophagy. Did you have a question?
Yeah, but with the units, I know we're like doing how you use it, so we'll talk about what I want.
Okay. Yeah, you were supposed to tell me about the time.
No, it's fine. It's just lunch after this. So.
Okay. Okay, well then.
Not just lunch. I mean, I'm really hungry.
Yeah. Enjoy your lunch. And thank you for the attention.
Okay. Thanks very much, everybody.
All right.